Receptors on the natural killer cell surface that regulate the recognition phase consist of two types. One is composed of the receptors that are homologous to the C-type lectins and is called the natural killer receptor complex. The other is composed of immunoglobulin-like domains called killer cell immunoglobulin-like receptors, also known as KIRs. These are shown on the natural killer cell surface as the forms KIR3D and KIR2D. However, the predominant control of the natural killer cell activation occurs through the C-type lectin receptor, CD94-NKG2 heterodimer. One complication in understanding the natural killer cell activity is that the same structural families, that is, the CD94-NKG2 natural killer receptors, contain both activating and inhibitory receptors. The NKG2 proteins differ for the inhibitory NKG2A and NKG2B, and the activation NKG2C and NKG2D protein structures of the CD94-NKG2 complex. Signaling by the inhibitory natural killer cell receptors suppresses the killing activity of the natural killer cells. The receptor for the target cell is the major histocompatibility complex 1, also known as MHC1. Natural killer cells will not kill healthy, genetically identical cells with normal expression of MHC1 as seen in other normal cells of the body. Therefore, the binding of the inhibitory natural killer cell receptor to normal MHC1 sends a negative signal to inhibit signals from the activation natural killer cell receptors, which are bound to the target cell activating ligand. However, if the MHC1 is 1, not dense enough on the target cell, or 2, if the target cell has processed antigen and displayed it in the pocket of the MHC1 illustrated here by the shaded red-yellow ball, or 3, if the conformation of the MHC1 proteins have been sufficiently altered, noted here by the stripped MHC1 proteins, then the binding of the altered MHC1 to the inhibitory natural killer cell receptor will send a positive to the activation natural killer cell receptor complexed to the target cell activating ligand. It is this positive signal that initiates the remarkable cytotoxicity. When the target cell, either a viral infected cell or cancer cell, displays an abnormal MHC1 and is targeted as non-self, the destruction begins. There are basically two types of destruction that take place. One is the release of cytotoxic molecules, such as tumor necrosis factor, also known as TNF, that are released from natural killer cell granule membranes that cause destruction of the cancer cell surface membranes. Another mechanism involves programmed cell death, termed apoptosis, that results in the destruction of the nuclear DNA of the cancer cell. Natural killer cells contain membrane vesicles called cytotoxic granules that package the enzymes that kill the cancer cells. The most important enzymes involved in the apoptosis event of cancer cell death are the pore-forming protein called perforin and a set of proteases called granzymes. The activation of the natural killer cells causes the release of these vesicles from within the natural killer cells, in which the contents are delivered directly into the surface of the target cell. The perforin proteins penetrate the cancer cell and create a cylindrical polyperforin pore through which the granzymes pass into the cytoplasm of the cancer cells. The process of cancer cell death initiated by the granzymes uses part of the same basic machinery that gives rise to programmed cell death, or apoptosis. In this application, one granzyme, granzyme B, activates a caspase, which then cleaves I-CAD, the inhibitor of caspase-activated DNA ACE. The active DNA ACE, having its inhibitor I-CAD inactivated, is now free to migrate into the nucleus and degrade the DNA. At this point, the cancer cell is destined for death, and the natural killer cell can migrate